We'd like to welcome you to the Sanguine Speaker Series webinar on PFE-001, a novel non-covalent modulator of hemoglobin, improves anemia and reduces sickling in a mouse model of sickle cell disease, presented by Kelly M. Nee, PhD. Kelly is a senior principal scientist in the Rare Disease Research Unit at Pfizer. But first, I would like to introduce Scott O'Donnell from Sanguine Biosciences, who has a few words about Sanguine. Scott?
Thank you, John, and thank you everyone for joining our virtual speaker series today, and thank you to Kelly Nee and Pfizer for speaking at the webinar. For anyone who does not know me, my name is Scott O'Donnell. I'm the Senior Account Executive for Sanguine Biosciences, and I handle most of the accounts in the Northeast and Midwest. Sanguine has worked with a lot of your companies, but for anyone who does not know us, Sanguine uses a direct-to-patient approach to procure minimally invasive samples directly from the patient's home via our mobile phlebotomists. We have a database of over 30,000 patients spanning various therapeutic areas, and we can work with very stringent IE criteria, as well as handling the IRB documents, patient consenting, and obtaining the patient's medical records for you folks. One researcher we've worked with over the years is Kelly Nee, who is, again, the speaker for today's topic. Kelly Nee, like John said, has a PhD, and she is a senior principal scientist in the Rare Disease Research Unit at Pfizer. She'll be talking today about sickle cell disease. And Kelly, thank you very much for volunteering to present, and we all look forward to learning more about this topic. So without any further delay, I will hand the stage over to Kelly to begin her presentation.
Hi, everybody. Thanks, everyone who's here to hear about PFE-001. This is our novel non-covalent modulator of hemoglobin that improves anemia and reduces sickling in our mouse model of sickle cell disease. Sickle cell disease is a neglected healthcare burden in the world at large. It's a well-understood disease. The first observations of the disease were back in the 1910s. In 1911, James Herrick observed sickle cells in a patient of his, and that was actually the first report of this disease in the literature. Later on in the 40s and 50s, the scientists identified both that this was really one of the first molecular diseases and that the cause of the mutation was this beta-6 glutamic acid devaline mutation. However, once we figured all of that out, there was sort of a lull in the research, and one thing that's lagged behind in this disease compared to other diseases that sort of have these initial genetic components is there really wasn't a lot of therapeutic options for sickle cell disease patients until the late 90s. One of the first attempts at a drug directly for sickle cell disease was tucarisol. That was in the early 90s. It wasn't until the late 90s that the transgenic sickle cell mouse was developed. And also in the late 90s, the FDA actually approved a drug called hydroxyurea for sickle cell disease. This is a repurposing of a drug that was actually synthesized first in the 1860s and was originally approved by the FDA for neoplastic diseases. Later, it was tested and found to be somewhat efficacious for sickle cell disease. And so that really became the first drug that was approved for sickle cells. However, in the recent most 20 years, there's been a great deal of additional interest in sickle cell disease. There's been interest both in curing the disease outright by gene therapy. Bluebird Biotech has done a great deal of work on curing sickle cell by correcting the gene error. And recently there's been some approvals of drugs directly for sickle cell that treat either the symptoms of the disease, such as vaso-occlusive crisis, and that would be Endari and crizanlizumab. And also voxelotor was approved in 2019, and that's a drug that really treats the root cause of the disease. So why are we so interested in this, and why do we still need additional therapies in this space is really because the incidence of this disease keeps going up. So we see that the distribution, as you can see in the map on the left-hand side of this panel, is it's really centered in sort of areas that are in the developing world. And in the developing world, we see that the incidence of this disease is only going to get larger. There's only going to be more patients who have this disease. So really, when we think about sickle cell, it's less of a rare disease in the mode of something like beta thalassemia or cystic fibrosis, and it's really more of a neglected disease. So the pathology of the disease is quite varied. The hallmark of the disease, of course, is the sickled cells. Those cells get integrated into, their cells get caught up in the circulatory system when they're in this sickled shape, and they cause occlusions, and that causes a great deal of pain. It causes a condition called vaso-occlusive crisis, where you get recruitment of inflammatory cells to that same space, and you get swelling, and it's quite excruciatingly painful. You get infarcts in various tissues, and when the infarcts happen in your brain, they can lead to a stroke. The sickled cells themselves tend to lyse quite quickly, so patients are chronically anemic. And if they get infarcts and occlusions in their lungs, they can get a syndrome called acute chest syndrome, which is quite dangerous, and one of the #1 causes of mortality among these patients. They can't clear their lungs, and they basically die of an acute pneumonia. Sickle retinopathy is another common manifestation of this disease. But really, the hallmark of the disease is the sickled cells. The sickled cells cause all of the problems, and really what problems happen to each patient is really very dependent on the patients. But overall, the life expectancy for these patients is quite low compared to a healthy person. In the developed world, the life expectancy for a sickle patient is 40 to 50 years. That's slowly increasing due to improvements in treatment in the developed world. In the developing world, however, the news is not as good. The life expectancy for a sickle patient in the developing world is around 20 to 25 years. And in the developing world, sickle patients under 5 are the most vulnerable. The mortality rate in Africa for patients with sickle cell under 5 is very high. It's actually the #1 killer of children under 5 in sub-Saharan Africa. So the root of the disease is a mutation in human hemoglobin. Almost everybody who's ever taken any kind of biochemistry class has probably heard about hemoglobin. Adult hemoglobin, which we call HbA, exists as a tetramer. It's 2 alpha and 2 beta chains. And it really cycles between these 2 conformational states that are related to the actual function of hemoglobin. Hemoglobin's main job is to carry oxygen from the lungs and deliver it out to the tissues. And so it does that by binding the oxygen at the hemes, which are shown here in these yellow ball and stick models. So the oxygen would bind sort of right where that orange dot is. And the difference between a hemoglobin that has no oxygens bound, which we call deoxyhemoglobin, and a hemoglobin that has all the oxygens bound, which we call oxyhemoglobin, the structural difference is quite profound. You can see that there from the sort of structure of the deoxyhemoglobin, when it transitioned to oxy, you can see there's a major conformational rearrangement. You can see the center of that protein opens up quite a bit. And that conformational rearrangement actually plays into the manifestation of sickle cell of the disease and facilitates the polymerization of the fibers. That's the hallmark of the disease. The mutation is a single point mutation. It's a glutamic acid to valine substitution on the surface. It's at the beta-6 position, and that results in sickle hemoglobin, which we call HbS. Other than that mutation, HbS and HbA are structurally identical. There's nothing wrong with HbS. It can carry oxygen just fine. What we have in the case of the mutation, the beta-6 Glu Val, it's really a gain of function mutation. So the mechanism of sickle cell disease is fairly well understood. And the polymerization that is the hallmark is driven by a series of hydrophobic interactions only when hemoglobin is in the deoxygenated state. So it's really driven by the presence or absence of these hydrophobic patches on the surface of the molecule. So if you look at my sort of simplified cartoon, hemoglobin, adult normal human hemoglobin, HbA, has no hydrophobic residues on the surface at all, so it's fine. Deoxygenated hemoglobin, because of that structural rearrangement I mentioned on the previous slide, a small hydrophobic pocket emerges, and that actually emerges on both of the beta chains, beta 1 and beta 2. I'm only showing it on one beta chain for sort of clarity in this figure. That's only present when the hemoglobin is in the deoxy state. Because there's only one hydrophobic region present, that doesn't present much of a problem. Oxygenated hemoglobin S, which is the hemoglobin that has the mutation, has a hydrophobic residue on the surface all the time. That's the beta-6, which is a little patch of hydrophobicity from the presence of that valine instead of the glutamic acid. Also kind of not a problem. The real problem comes in when we have deoxygenated HbS. In that case, we have both this little corner pocket that was the result of the structural rearrangement, and we have one of these beta-6 patches. We have one of each on each beta chain, and those pockets and patches can actually associate with the pocket or patch of another hemoglobin tetramer, and the way that those are arranged is they sterically line up in such a way that they can form this long polymer. It's a very straightforward sort of hydrophobic interaction that unfortunately has this terrible consequence of eventually what happens is the cells get packed with these fibers. Hemoglobin's at a very high concentration inside the cells. They get packed with these fibers, and they adopt this format that we see, the sickled shape. That's sort of the visual representation of the disease. We know a lot about how these fibers form inside the cell. We know that the whole process starts with a few hemoglobin tetramers coming together into what we call a critical nucleus. And so this process of coming together and coming apart and coming together is known as critical nucleus formation. And the time that's required for that is what we call the delay time. And what we know about it is that it's extraordinarily concentration dependent. So the higher the concentration, the faster that this nucleus is going to form. And once you have a critical nucleus, the formation of a whole polymer and actually additional polymers which build off of that initial polymer goes very quickly. And at that point, it's almost too late to stop the process. So the idea is, because nucleation occurs only in the deoxy state, and we know that when cells get back to the lungs to be reoxygenated, this whole process resets. The fact that it's highly concentration dependent and it only occurs in the deoxy state means that we can see a small change in concentration will then translate to a very large increase in the time that it takes to form this critical nucleus. And really all we have to do at that point is beat the time required for the red cells to get back to the lungs to reset the process, and we would actually wipe out the formation of this critical nucleus. And if we could just prevent that nucleus from forming, we could prevent the polymer from forming, and therefore we could prevent red blood cell sickling and ameliorate most of the disease pathology. So our idea is, and what we've done with PFE-001 is we've designed a molecule that stabilizes the oxygenated state of HbS, which we know cannot polymerize and thus will not manifest the disease state. This is sadly not a new idea. I wish that I had thought of it myself, but I did not. Stabilizing the oxygenated state by a kinetic approach is something that's been attempted a number of times. The idea is very simple. You stabilize the oxygenated state. As I mentioned, you inhibit sickling. If you don't have sickled cells anymore, you won't have anemia. You won't get vaso-occlusive crises. You won't end up with end organ damage. That's also very prevalent in older sickle cell patients. And you would basically cut off the chain of the disease, and you would have what would be representative of a substantial improvement in the medical quality of these patients' lives, as well as the overall quality of their lives. Pardon me. This has been tried before, starting in the mid-70s. A group of physicians tried extracorporeal carbamylation. So in that case, they actually extracted some percentage of the blood from a sickle patient, around 20%, and they modified the hemoglobin by carbamylation. And then they reinfused it into the patients. And what they found was if they did this for a number of weeks, they did it for by about week 3 of this treatment, they saw substantial improvement in anemia. They saw about an 80% reduction in sickle cell crisis. They saw about an 85% reduction in hospital crisis days. So these patients were profoundly improved in terms of the amount of time they were in some sort of crisis. They also saw complete healing of the chronic ulcers. So this therapy was quite successful. The only problem was the method of administration was extremely onerous, and it was not really feasible for a great deal of the world that is impacted by this disease. So that approach was eventually sort of abandoned. However, the idea remained, and later on, starting in the 90s, the reactive aldehyde compounds started to come to the front of the line. These are compounds that bind the hemoglobin alpha chains and stabilize the oxy state, and they do that by forming a Schiff's base with the N-terminal valine of the hemoglobin. One of the first attempts to go into the clinic was a compound called tucarisol. This compound showed significant improvements in hemoglobin concentration and anemia. Unfortunately, we couldn't get a better look at what else this drug might have been able to do because the trial was ended early due to safety concerns that were in fact linked to the reactive aldehyde. Recently, voxelotor was just approved for the treatment of sickle cell disease. This is also a compound that has a reactive aldehyde. In the case of the phase 3 trial for voxelotor, 59% of the subjects showed a 1 gram per deciliter increase in hemoglobin. That was their endpoint, so they met their endpoint. They saw significant improvements in other markers of hemolytic anemia. The study wasn't really powered for it, but they didn't see much of a change in VOC. And as I mentioned, the compound was approved in late 2019. It was clear from the experience of tucarisol and voxelotor that it was possible to show improvements in the hemoglobin and other markers of anemia by using a small molecule. However, learning from the experience of tucarisol that reactive aldehyde does in some way can in some way pose a safety risk, we decided that we would attempt to discover a non-covalent molecule that could do the same thing, that could bind the hemoglobin, stabilize the oxy state. And what we came up with was PFE-001. It is, as I said, a non-covalent binder. It binds ditopically, so 2 molecules per hemoglobin tetramer. It stabilizes the oxygenated state and shifts oxygen affinity, and I'll show you some data on the next few slides that speaks to that. And as of now, we see that it has a permissive safety profile for FIH trials that we're planning on getting into later in the year. But before I get to the data, one of the things that we have learned both from the experience of carbamylation and from the experience of voxelotor is that when you're stabilized in the oxygenated state, the coverage level is extremely critical for efficacy. So GBT's preclinical data, which I'm showing on the left-hand side of this slide, showed that mice that were dosed with voxelotor, which at the time they were calling GBT 440, showed quite a bit of variability in hemoglobin occupancy at the same dose. So you can see that the 2 groups of animals that were dosed, they saw, with the same dose, they saw occupancies between 11% and 40%. And there wasn't a really great explanation for why that was. But this variability was quite apparent, and that actually carried on through their human trials into their phase 3 data. I'm showing on the right-hand side the waterfall plot from their phase 3 paper, showing that their 59% of their patients who were taking voxelotor at 1,500 milligrams showed an increase of hemoglobin greater than or equal to 1 gram per deciliter. And at 900 milligrams, 38% showed an increase in hemoglobin of 1 gram per deciliter or greater. But what you can see from these waterfalls is that there's a great deal of variability in how much 1 gram per deciliter or more that they got. There's patients who did very, very well and saw increases of upwards of 3 to 4 grams per deciliter. And then there were patients that just made it to 1 gram per deciliter. And this is all in the background of the same dose. And so that variability that was apparent early on still seems to be carrying through to their human data. And if we contrast that with the experience of carbamylation, in those studies, carbamylation, patients who were carbamylated saw an average increase of 2 grams per deciliter, and they saw about 30 to 50% coverage of their red cells. And so it's very clear that the better your coverage, the better your response, and the more robust your increase in hemoglobin, the more likely you are to have improvements in these other factors that are important, such as a vaso-occlusive crisis. As of now, the clinical benefits of rising hemoglobin haven't really been established for voxelotor or any of the other potential therapies. However, I know that they are in the middle of doing those studies to show that linkage, and I'm sure we'll be hearing from them shortly. So back to PFE-001. This we took this in a very holistic way. One thing Pfizer's very good at is screening small molecules. So we started with an affinity selection mass spectrometry screen using purified human HbS in the oxygenated state. One of the reasons that we chose ASMS over other options was that this particular method explicitly excludes covalent modifiers. So we wouldn't have been able to find a covalent modifier if we tried. And we actually did put them in as tests and we never picked them up. So all we were ever going to find from this method was going to be non-covalent modifiers of hemoglobin. So from that initial screening set that had around 750,000 molecules in it, we got around 350 hits. And from those hits, we synthesized about 2,500 additional molecules to optimize the sort of PK/PD properties. And what we ended up with as our clinical candidate was PFE-001. Up here in the top right is a crystal structure of PFE-001. You can see, as I mentioned, it's a ditopic binder. So the two molecules bind to each hemoglobin tetramer, and they actually interact with each other. We know that this compound preferentially partitions into red blood cells. It significantly stabilizes oxyhemoglobin in vitro and in vivo. And I'm showing on the bottom right-hand side, it shows coverage-dependent inhibition of HbS polymerization. So as we increase the coverage or the occupancy, we see an increase in the percent change of delay time. At some point, you change the delay time so much that there's nothing left to change and it goes to zero. So we had very favorable in vitro data that suggested that this compound could be our guy, and it was time to bring it into an in vivo study. So that was our next step. So we're fortunate to have a really great mouse model. The Towns mouse has human hemoglobin in this human hemoglobin S instead of mouse hemoglobin, and it recapitulates very well a number of the features of sickle cell disease in humans, in particular the hemolytic anemia markers. And so we see that this particular mouse has a very low hemoglobin relative to the control, and that's mimicked by what we see in human patients. Similarly, human patients have very high reticulocyte count, as do the Towns mice, and they have very low red blood cell numbers, and that is also mimicked by the Towns mouse. And so it's a very good model for sickle cell, and we thought it was the right choice for doing our next set of studies in vivo. We started with some single doses to understand our PK a little better. We started with doses of 10, 40, 220, and 490 mg per kg, and you can see in the table on the top of the slide. And what we saw was quite interesting. As we increased the dose, we increased blood concentration and blood binding, so our pharmacokinetics are actually nonlinear. As we see these increases in blood binding, our clearance actually decreases, and so that results in an increase of our half-life as we increase the dose. And this half-life sort of increases until it plateaus with concentration in the blood of around 2 to 4 millimolar. So it's not forever, it's not infinite. We see that it plateaus at around 2 to 4 millimolar, which is less than the total hemoglobin concentration. So we don't think that there's a scenario where we could bind all the hemoglobin, even with these favorable sort of nonlinearity of our pharmacokinetics. Once we've done our single dose, we were able to hit on a dose for a chronic study, and for that we chose 200 mg per kg twice daily. This is an oral study, and we dosed those animals for 15 days. And what we saw was that PFE-001 achieved very high levels of exposure, around 4.2 millimolar by day 5 of dosing. And we maintained that coverage throughout the duration of the study. And we had originally chosen the 200 mg per kg as twice daily as our dose in order to achieve around 20 to 30% target coverage. We were trying to sort of mimic what we had seen in previous articles and what they had seen in the carbamylation paper. And really what we got was exposure that approached 60% coverage. So we got extraordinarily good coverage, and from a dose that's not even at the top of the dosing that we did in our single dose study. So that was excellent news for us. And what does all of that coverage get you? It gets you a very apparent stabilization of the hemoglobin oxy state in vivo. And so on the top left, I'm showing two curves. The green curve is PFE-001. These are examples from our treated animals, and the blue curve is the vehicle. And you can see that there's a large change in both the P50, which is the point at which 50% of the hemoglobin is oxygenated and 50% is deoxygenated, and the P20, and that's the point where 20% is oxygenated and 80% is deoxygenated. You can see there's a large gap. And when we quantified that, across all of our animals, we saw that 001 shows a 53.7% reduction in P50, that's on the top plot, and an 84.4% reduction in P20. And these numbers really suggest that we're seeing a significant modification of our HBS. And recall from the few slides, a few previous, that we know that coverage is extraordinarily important to getting really good efficacy in these animals and presumably in patients. And so we were very excited by these results. We also saw a substantial reduction in red blood cell sickling. In these experiments, blood was drawn from either the vehicle animals or our PFE-001 treated animals, and it was subjected to hypoxia for 4 hours, and then we stained and imaged and then did quantification of the sickled cells. And you can see in the vehicle, in images from the vehicle, which are on the left, there's quite a few sickled cells. It's very apparent that there's a lot of sickling in those animals. And you can contrast that with the images on the right, where we see PFE-001 treated animals. You can see a lot more healthy cells, far fewer sickled cells. And when we quantified that across all of our animals, we saw a 37.8% reduction in sickled cell. And this is under a hypoxic exposure that's quite stringent compared to what actually would be going on in vivo. In addition to the impacts on oxygen affinity and red blood cell sickling, we saw that PFE-001 significantly impacted important markers of hemolytic anemia and also impacted some markers of vascular inflammation. So at the end of our 15-day dosing with PFE-001, we saw a 42.4% increase in hemoglobin. That's shown at the top. You can see that our treated animals actually started to approach the wild-type black 6 levels of hemoglobin, and the total amount of hemoglobin that was gained by these animals was about 5 grams per deciliter on average. And you can see the contrast between it basically doubled the amount of hemoglobin that the control animals had. Other markers of hemolytic anemia also showed significant changes. When we look at our red blood cell count, we saw a substantial increase in RBCs, and that actually got us into the normal range for the black 6 animals, as opposed to the untreated animals, which sort of stayed very low. Similarly, reticulocytes, which are a marker of sort of cell production, you can see we had a very significant decrease in retics, not quite to the baseline of a healthy animal, but substantially improved over what we saw in the untreated animals. In addition to what we saw for hemolytic anemia, we tested a few of the markers of vascular inflammation. The study wasn't really powered to do that, so this was sort of something we tried just to see if we could see anything. And in fact, we did. One of the things that we were very pleased to observe was that PFE-001 shows a small but still significant statistically change in soluble VCAM, which is a marker of vascular inflammation. And so we were hopeful that this is sort of pointing us in the direction of the fact that we have this very high coverage and we have this improvement in hemolytic anemia is going to translate into improvement in the vascular inflammation that then would translate into improvements in people in VOC and other inflammatory episodes, which would be sort of very clinically meaningful for these patients. So that's sort of the story of PFE-001. In conclusion, we have previous reports that suggest that compounds which bind in the alpha-1 valine pocket of hemoglobin are able to impact both hemolytic anemia and VOC if we can get that target occupancy. They really need a lot of horsepower in order to see those impacts on VOC. PFE-001, we hope, has the muscle to do that. It's a potent, specific, and efficacious non-covalent modifier of HBS. It shows consistent pharmacological effects at the exposure levels that we saw. It decreases RBC sickling by about 37.8%, and it positively impacts markers of hemolytic anemia, including a 42.4%, which is a 5 gram per deciliter increase in hemoglobin. And based on these strong in vivo data, we have plans to advance PFE-001 to the clinic. And I see there was a question that when are we going to further studies or in the clinic? And the answer is now. We have a phase 1 first-in-human study planned. The design of that study is a single and multiple ascending dose of PFE-001 in healthy adults. It's going to be a double-blind study with sequential dose escalation. And really what we're looking for in that study is obviously safety. We're going to look for adverse events, of course. We're also going to look for changes in vitals, ECG, and our lab test findings. Obviously, the other thing we're really interested in with this molecule is pharmacokinetics after single and repeat dose. And then for our pharmacodynamic effect, we're lucky enough that we can actually read out a PK and PD effect in our healthy volunteers, the change in oxygen affinity. So we're going to look for modulations of P20 and P50 for that study. And then, of course, we're going to also look at some differences in food, plus or minus food, and a few different drug formulations is really the plan for that study. So I'll stop there, and I'm happy to answer questions which I think are going to come in over the chat, and they're already coming in. So the age range of the study for this phase 1 will just be adults. We are in discussions about pediatrics, but we start with adults for our healthy volunteer study. I also see that somebody asked how long this research took. I think as of now, from sort of day 1 to where we are now was about, I would say, 5 to 6 years of sort of intense bench research, and subsequent to that, all of the other things that needed to go on with other groups and you know, safety evaluations and pharmacokinetics and formulations and everything. And how many people are on my team, I'll show you. I hope. I'm going to try. Okay, so this is the team. It's quite large. There's no way that something like this could be accomplished without the contributions of an enormous number of people, and every single one of them made a huge contribution. You can see that this is a large collaboration across multiple departments at Pfizer. This is really a whole company effort, at least worldwide research and development. I think there's people on the team from every single part of WRD, and now that we're getting ready to go to the clinic, we have even more people from different areas. So this covered people from Cambridge, New York, Groton, Andover, name a site, name a Pfizer site in the United States, and we probably have someone from the team who's based there. So it's a huge collaborative effort. So I hope that answers everyone's questions about how we did the research. How do we think we can differentiate from voxelotor? I think, we have some ideas about that, but we're not at this point really prepared to discuss that without, we need to see the results of our phase 1 trial before we can really speculate on how we might differentiate from voxelotor. Another question, did we see improvement in organ damage in the in vivo studies? We did not, because we didn't look for that. We think that 2 weeks is probably not a sufficient amount of time to see organ studies. When and if we have another study with this molecule, we would take it out a little bit longer and see if we could see organ damage. How do we measure affinity of this drug to hemoglobin? I can tell you that it is to the intact tetramer. We're saving the method for our paper, but it'll come out soon. But we do measure the intact tetramer. It's not just a single chain. Yeah, so the KD listed was for the intact tetramer. We're doing a multiple dose study, so we haven't been able to predict. We're not predicting yet. We're not saying what we're predicting for our dose that's efficacious in patients. The question was, what dose do you predict will be efficacious in patients? We're going to wait and see. And we're not, the question is, how long will this go through the clinic? I don't think we know the answer to that yet. Okay, it seems like there's no more questions. So again, thank you to everyone who participated and came to this talk. I appreciate it. Oh, one question, BID or QD? The study that we did in the animals was twice daily. As for our in human studies, we aren't talking about that quite yet. One more question, do you plan to combine with other agents on the market? Everything's on the table at this point, but we haven't had any specific discussions about that. One more. The reduction in inflammation is very encouraging. Any sense of what the driver is? Not really. No, we haven't. Like I said, this was a sort of minor effect, so we didn't have a great deal of opportunity to really look at it. Obviously, the less hemolysis you have, the less likely you are to have, sickling, or sorry, the less sickling you have, the less likely you are to have hemolysis. You're not going to have free heme. You're not going to have sickled cells sort of cutting up the circulatory system, and all those things are going to contribute to inflammatory responses. Okay, I'm being assured that that is it. So again, thank you very much to everyone who attended. I appreciate it. And I hope everyone is staying healthy and is going to have a lovely rest of the day.
Thank you, Kelly. And thank you all for joining Sanguine for our S3 webinar. PFE-001, a novel non-covalent modulator of hemoglobin, improves anemia and reduces sickling in a mouse model of sickle cell disease. For a list of upcoming webinars, please visit researcher.sanguinebio.com. If you have a need for patient samples for your research, please visit sanguinebio.com. Thank you again and enjoy the rest of your day.